Yeah, good day, everybody. Thank you very much for letting me start off the session. So it differs quite heavily from what we've heard so far, and this talk is about the acquisition part of layer-dependent fMRI, especially the contrast mechanisms, and I want to focus on the sensitivity and specificity aspect of those. So first of all, let me define those terms. With sensitivity, I mean the ability to detect functional signal changes in general. And this is, of course, proportional to the functional contrast-to-noise ratio. With specificity, I mean being sensitive to the signal changes at the locus of activity, i.e. the neurons. But fMRI in general only measures activity indirectly by tracking changes in the hemodynamics, so changes in blood flow, blood volume, and oxygenation. Hence, it is imperative to look at the Vaso architecture. And in case of the primal visual cortex, this is a schematic showing that it's actually quite organized. So oxygenated blood comes from the pili arteries and goes through these diving arteries, which branch into smaller and smaller arterioles, and then finally into the capillary bed, where oxygen gets exchanged with the parenchyma. And then deoxygenated blood goes basically the other way around, following these venules which merge into ascending veins and then into the pili veins. And then talking about specificity, one can look at how the cells are organized in their stereotype layer fashion, and you can look at the microvasculature, which kind of closely resembles the cell density distribution. And moreover, it also resembles the oxygen metabolism. So this is a Cox staining that you can see as soon as the density increases, the metabolism also increases. So the bottom line of this slide is you want to be sensitive to whatever happens at the microvascular compartment. But the bolt response is actually a result of many things happening in a cascade of events. So it starts with the neural response, and then blood flow increases, blood volume follows, deoxyhemoglobin decreases, and then you're at the bolt response. So in order to be specific, people also try to measure earlier in this chain to be closer to whatever happens at the neuronal level. Now, you can broadly classify fMRI methods in grand echo bolt, spin echo bolt, and non-bolt techniques, where the workhorse sequence is really grand echo EPI because it has this unrivaled temporal efficiency. For spin echo bolt, there are several types like spin echo EPI, balanced steady-state free precision, 3D gradient and spin echo, T2 preparation. And in non-bolt, there's Vaso EPI, maxation transfer or arterial blood contrast, arterial spin labeling, calibrated fMRI. And at this point, I would like to make a disclaimer. It is impossible for me in 20 minutes to cover all the methods in the detail I would like to. So I want to focus on these methods to highlight the sensitivity and specificity aspects between these these three main classes. So when looking at gradient and spin echo bolt, we need to look at what is happening inside the vessel, so the intravascular effects, and what happens outside the vessel, extravascular effects. And I want to start with the extravascular effects first. It has been shown in experiments, but also in many simulation studies, that when you look at the bolt sensitivity as a function of vessel radius, then the grand echo bolt is very sensitive to whatever happens at the capillary compartment, very small vessels, but it's also very sensitive to what happens around big veins. Whereas in T2-weighted sequences or spin echo bolt, these tend to be more, like those single changes, tend to be more specific to whatever happens at the microvascular compartment. So if you would do a high-resolution grand echo bolt 7 Tesla imaging, you would still be very sensitive to those big vessels, which by looking at the Vaso architecture, you know that they are more away from where you're actually interested in. And moreover, it's not only that the pili vessels are at the pili surface, but they also degrade the specificity when you go deeper into the parenchyma. And this is shown here by Vaso et al., where you can see here in these red areas, with depth 1, depth 3, depth 4, and this is a normalized signal as a function of Euclidean distance to this large vein. The Euclidean distance is shown here in these isocontours. That you can see, for instance, that at depth 3, like here, you are sensitive to signal changes which are 1.5 or 2 millimeter away from this large vein. So this vein affects what you measure in mid-gray matter. And yeah, this is a problem for gradient echo bold. Now, I want to explain briefly why spin echo bold is more specific compared to gradient echo bold. What you can see here is a capillary and a vein, and due to the deoxyhemoglobin concentration inside the vessel, there's a field prepudation which has this dipolar shape outside of the vessel. In black here you see a diffusion path of the proton, and typically the diffusion path would actually be the same, and the vein would be 100 times bigger, but in order to now not blow up the screen, the vessels are scaled identically, but the diffusion path is made smaller. Now, if you track what kind of dephasing or field inhomogeneity this proton experiences while it is traversing this field change, then it experiences a field variation which is of a random fashion. So if you would apply a 180-degree focusing pulse, which is done in spin echo imaging, you would not be able to refocus whatever happens to a spin diffusing around the capillary. If you look at the vein, then this proton here experiences not so much field variation. The field variation appears to be static from the point of view of this proton. So if you would apply a 180-degree pulse at your time of echo, you more or less perfectly refocus signal variations for this proton. So in spin echo bold, you made yourself sensitive to whatever happens outside of the capillary. Now, looking at the intravascular effects, this is an Indian ink stain of the primal visual cortex in human. Here's white matter, here's the peel surface. And if you would look at the baseline blood distribution as a function of cortical depth, this would look like this. And if you would be sensitive to the intravascular signal change, then you would see, you would experience again this peel surface bias. However, we typically do layer-dependent fMRI at ultra-high fields because of its higher baseline SNR and higher bold effect in general. And if you would make yourself sensitive to the gray matter signal change by choosing a TE which is roughly equal to the T2 star of gray matter, you reduce your sensitivity to venous blood signal changes because venous blood has a very short T2 star. Especially it gets shorter and shorter the higher the field strength become. For spin echo bold, this is the intravascular signal as a fraction of the total fMRI signal ratio. It tends to be the same, although the T2 times of blood are in general longer than the T2 star times, meaning that, for instance, at 7 Tesla, we are at this magical point where you have the microvasculature and the macrovasculature contributes roughly equally to the total fMRI signal. If you increase in field strength, the microvascular distribution becomes higher relative to the macrovascular distribution. So in spin echo, you still are sensitive a little bit to intravascular signal changes. Now coming to the implementation, as I said, the workhorse is echoplanar imaging, where instead, in case of gradient echo EPI, you sample, your acquisition window looks like this, and you sample this T2 star decay. When you look at spin echo EPI, then only at the center of K-space you have perfect T2 weighting. And depending on your window length, you get more and more T2 star weighting in outer part of your K-space. And because we are acquiring bigger and bigger matrices, our acquisition window becomes longer and longer and longer, hence our T2 star weighting becomes also more severe. One way to circumvent this is to use segmented acquisition, where you repeat excitation and readout, and then only acquire portions of your actual readout window, and then put everything together in the end. And a very influential study performed by Göns and Logothesis have looked at the segmentation factor and how it affects the layer profile in spin echo EPI. So what you can see here is high-resolution macaque data, 0.5 millimeter isotropic resolution, a signal change as a function of cortical depth. Here's the peel surface, here's white matter, and this is for a full-fold segmented spin echo EPI. So the acquisition window is roughly 30 milliseconds. Now, this is for 8-fold segmented acquisition. The peak gets heavily reduced. This is for 16-fold segmented acquisition. And here you can argue, okay, I'm not so much affected by this peak anymore. But a 16-fold segmentation has a detrimental effect on your temporal efficiency because you need to repeat the acquisition basically 16 times to get one single volume TR. And in a contrast investigation study, using T2 preparation, we have seen similar things. So what you can see here is functional signal change as a function of cortical depth. Here's white matter, the gray box is gray matter, this is CSF, and we varied the T2 star times and the T2 preparation times. So this is like increased T2 weighting, increased T2 star weighting. And what you can see is that if you extrapolate to a 0 millisecond grained echo TE, so no T2 star dephasing happening, then you are nicely sensitive to the signal changes inside the parenchyma. At somewhat lower TE, you see that you get some signal change here, which is due to the higher intravascular signal. So you need to have a bit longer prep echo times or spin echo times. But you can also see that as soon as you introduce some T2 star weighting, only 2.3 milliseconds, you start to see a signal change at the peel surface, which increases with longer echo times. However, our data also suggested that this peak here is actually coming from extravascular effects in CSF. And CSF has a very long T2 and T2 star compared to other tissue types. And this you could, in principle, address by going to higher spatial resolutions, because you're only interested in the gray matter, right? And you see the signal change inside CSF only through partial voluming because your voxel covers gray matter and CSF. So you could address this in principle by going to higher spatial resolutions. Now, an alternative method to sensitize your signal change more specifically is CBV weighting. And the idea is that if you have a voxel containing gray matter and a network of capillaries and a large vessel, then upon activation, the capillary system tend to dilate more compared to larger veins. So if you would make yourself sensitive to this change, you would make yourself sensitive to microvascular signal changes. And one way to do this is use vascular space occupancy, which is essentially a blood nulling technique. So you null the blood, and then upon activation, this increases the blood volume, and you get a negative functional contrast. In the early implementation, it was not so much possible to get a large field of view. So the imaging field of view is this blue line here. It's only 7 slices with 0.8 millimeter slice thickness. The red area here is the saturation region. This diagram down here belongs to a newer implementation of Vaso, where you can increase the coverage by a factor of 4, roughly. But the general idea stays the same. Namely, you apply a global 180 inversion pulse, so all the longitudinal maxillation gets inverted, and then you capitalize on different T1 relaxation behavior between gray matter and blood. So at some point, you reach this magic point where blood maxillation is 0. And here's where your center K-space is acquired. These tiny blobs here are EPI readout kernels. So you're acquiring, and at the center of K-space, you get your blood nulling condition. Gray matter exhibits one signal. But because this is EPI, remember, during your EPI readout, you get T2 star weighting. So you need to account for this. Because the bolt signal is positive and the vaso signal is negative, it would cancel your precious vaso contrast. And in order to do so, you need to acquire a helping volume without a 180-degree pulse. So here you get both gray matter and blood signals, and you hope that the T2 star contribution in these two volumes is roughly identical, so you can dynamically divide those two on top of each other and get only the CBV change. In a recent implementation of Vaso called the Magic Vaso, it does not capitalize on blood nulling, but on simply blood saturation, which greatly increases the efficiency. So the 180-degree pulse is gone, and you keep on acquiring the entire time, with the difference that in odd TRs, odd volume TRs, you acquire your signal at a very low flip angle. So the signal from blood and gray matter is not altered that much. They are more or less on top of each other. Whereas in even TRs, you acquire your signal at very high flip angles. So then you again capitalize on the T1 differences from gray matter and blood, and you get a blood signal suppression relative to the odd TRs. And with that, you can increase the coverage by a lot, but you also reduce the sensitivity and specificity because it's not a blood nulling technique anymore, it's only a blood suppression technique now. This is an example showing what CBV based off acquisitions can do. 0.5 isotropic resolution. It's a segmented EPI. And you can see that the CBV change nicely follows the gray matter ribbon. This is, for reference, a high-resolution flash showing these nice intercortical veins and these big peel veins. This is the stria of Gennari, this dark band here. So CBV is more specific. The bold signal tend to be more blobby, and you have this massive signal change at the peel surface where the vessel is located. And if you look at the signal change as a function of cortical depth, you see that bold tends to have this bias towards the peel surface, towards the CSF compartment, whereas Vaso is more confined to gray matter. Now, alternatively, you could use a technique called maxation transfer weighting and achieve a CBV weighting slightly differently, where you opt to reduce the gray matter signal and leave the blood signal untouched. Hence, you would get a positive signal change upon activation associated to the dilation of the capillaries. And in addition, if you combine this with an gradient echo readout, you would reduce yourself to the sensitivity to extravascular effects, which you know are less specific in gradient echo bold because you reduce the gray matter signal. And we have investigated this contrast similarly to the T2 prep paper I've shown earlier, where we compared MT on versus MT off in a layer fMRI experiment using different echo times. And you can see here that these lines are closer together compared to this, which reflects less extravascular signal change, which is good. And you might also see that this line here is a bit higher, or like these two, blue and green, a bit higher compared to this. And this can maybe be appreciated better by looking at normalized signal changes, where I normalized the signal change to the peak of the bold response. So here's everything at normalized to 1, and they are more or less on top of each other, whereas here you can see a significantly higher signal change in gray matter for the 0th and the first echo time, reflecting CBV sensitivity. And I would like to end this talk by showing you this figure and give you some advices, if I may, what kind of sequences, contrast mechanisms you want to choose with the question in mind. So in layer-dependent fMRI, we always need to balance between these three regimes: high coverage, high speed, high spatial resolution, and this is underlined because it's not only the voxel size, but also the intrinsic resolution given by the MR contrast. And if you want to have high coverage and high speed, then T2 star weighted bold is the only way to go, actually, because it can be implemented so time efficiently. You can be very quick and have a large coverage and higher resolution, and then you need to deal with the venous bias differently, for instance, by designing clever paradigms, which I think will hear in a Hahn lecture. Alternatively, if you can afford a lower coverage, then you can go for T2 weighted sequences, like 3D Grays have shown very nice results for small coverages. If you can afford the double TR for Vaso, then you can reduce your temporal efficiency a little bit, but still having nice coverage and nice resolution, spatial and intrinsically. And with that, I'd like to thank you very much for your attention.
So, thanks very much for that overview. Do we have any questions? I think your conclusion slide was a bit of a spoiler for what is possibly to come. But
yeah.
Yes.
Yeah.
Yes, please. So we've got a question. Can you run with the microphone?
Just one simple question. Do you implanted the parallel imaging using this sequence, or no, you just use the same, like the standard sequence? Because if you go to parallel imaging, you can reduce the temporal resolution. You record the whole brain, divide it into different sections, and then you fasten the recording. Then T2 weighted imaging also will be really fast. Do you do the same as parallel imaging, or no, you just use the standard?
Well, you can consider parallel imaging as being standard these days. So you always do an acceleration factor of at least 4. If you have a whole brain coverage, you can play with certain variants of a method called caipirinha. I don't know if you're familiar with it. you can increase the total acceleration factor by to a 8-fold, I think, is reasonable. But even if you can basically only increase your acceleration by increasing the coverage, because you need to exploit the coil sensitivity variations, right? And because you increase your coverage, you have a higher coverage, but you invest all your acceleration capability in making your TR compatible with standard TR. So I'm talking about TRs of 2 to 3 seconds. And this is only done with gradient echo EPI. As soon as you start to use your spin echo, you would need to decrease your acquisition window even further to account for this T2 star contamination during your readout. And it's really like the readout length which is the killer.
Yeah. Good. Thank you.
Yep.
Are there any further questions? If not, maybe